Welcome all. Welcome once again in this video. So we are going to discuss about the structure or morphology of paramecium. So this paramecium is a microscopic organism, elongated body, and this paramecium is often described as slipper shaped organism or having spindle shaped body. This organism is described as a slipper shaped organism due to their structure or having spindle shaped body. The body of this organism is elongated and rounded at their anterior end. The anterior end is blunt and rounded as compared to the posterior end, and this posterior end is somewhat pointed. Anterior end is blunt or anterior side of this organism is rounded, while the posterior end is pointed and having a slipper shaped like structure. The body is distinguished into oral or ventral surface and aboral or dorsal surface. The oral surface of the body is called as ventral surface, that is the ventral surface, and aboral surface is the dorsal surface of this organism. So this is having a, when we see the structure, we can find there is having an oral groove. The ventral surface of the body, this oral groove is found, which is a prominent and a shallow depression. This oral groove is a prominent and shallow depression. This is a depression which is called as oral groove. It is a shallow depression and it originates from the middle of body and extend to the left side of anterior end. Extend to the left side of the anterior end, and posteriorly this oral groove leads into a deeper conical structure that is called as vestibule. It is oral groove, then posteriorly leads into vestibule, which in turn forming a cavity that is called as buccal cavity. Buccal cavity having again this buccal cavity lead into a structure, particular structure that is called as cytostome. This cytostome is a mouth like structure, so called as cytostome. Then this cytostome leads into a particular structure that is called as cytopharynx. That is the gullet or cytopharynx is the next posterior structure of the cytostome, and the cytopharynx at its proximal end forming food vacuole. Then such a type of food vacuoles, here we can find many food vacuoles. These food vacuoles are found scattered in the cytoplasm. These food vacuoles are formed in this particular structure by this buccal cavity and cytopharynx forming food vacuole. So oral groove lead into vestibule, vestibule lead into buccal cavity, and then forming the cytostome and cytopharynx, and the cytopharynx then forming and lead into food vacuoles. The food vacuoles are formed. All these structures are called as oral apparatus. That is, oral groove, vestibule, buccal cavity, cytostome, cytopharynx are called as, these particular structures are called as oral apparatus. So such type of oral apparatus are found in the paramecium. Then the overall body or the external envelope of the body is called as pellicle. So the overall body is covered by particular type of envelope. Body envelope is called as pellicle, which is clear, firm, and elastic cuticular membrane, that is called as pellicle. Pellicle is the outermost covering of the body, and this pellicle is protect or cover the entire surface, body surface of this organism. This pellicle is clear and firm type of membrane which covers the surface of this organism. Then we can also found that the overall body is covered by small, entire body is covered by, or entire surface of the body is covered by numerous, that is many small hair-like fine projections, that is called as cilia. So entire body surface is covered by tiny hair-like projections, that is called as a cilia, and this cilia is about 10 to 12 micron in length and 0.27 micron diameter. They are having 10,000 to 14,000 cilia, 10,000 to 14,000 cilia covering the whole body surface of this paramecium. And then their length is uniform throughout except towards few longer cilia found at the posterior end of the body. Their size is uniform all over the body, but the cilia forming or the length of the cilia are found elongated towards the posterior end of the body of this organism. That is the posterior, towards the posterior end of the body of this organism, this cilia becomes elongated and forming caudal tuft of cilia, due to which this specimen having name chordatum, that is the paramecium chordatum. Then about the cytoplasm. So cytoplasm, within the pellicle, the cytoplasm of this organism is clearly differentiated into two regions. Cytoplasm is clearly differentiated into two regions. The outer narrow peripheral clear dense zone is called as the ectoplasm. Cytoplasm is divided into ectoplasm and endoplasm, differentiated into ectoplasm and endoplasm. Ectoplasm is the narrow peripheral zone layer, peripheral clear zone. Then endoplasm is found towards the inner side of the body. This endoplasm is a larger central granular semi-fluid zone. Endoplasm is a large central granular semi-fluid zone, or the middle, which covers the middle portion of the body. It includes many cell organelles like mitochondria, Golgi bodies, ribosomes, food granules. All these material can be found in that endoplasm. So cytoplasm is divisible into ectoplasm and endoplasm. Then there are some structures that is called as trichocyst. These trichocysts are rod-like structures. Where these trichocysts are rod-like structures. These are having many such a rod-like structure beneath this pellicle, that is called as trichocysts. These trichocysts are rod-like or oval organelles present throughout the ectoplasm and with basal bodies and oriented at right angle to the body surface. These trichocysts are oriented towards the right angle to the body surface. This can measuring about 4 micron in length. The length of this trichosis is about 4 micron, and each trichosis consists of an elongated shaft-like projection. This trichosis having such a type of elongated shaft-like projection, elongated shaft-like part, and terminal part of this trichosis is pointed, having a pointed tip called as spike. This pointed tip of this trichosis is called as spike. So they are having two structures of this trichosis, that is, this is a broad region, broad part. This broad elongated part is called as shaft, and a terminal pointed structure that is called as tip. These two structures are found in this trichosis. But the function of trichosis is not well known. The function of trichosis is not well known, but it is believed that this trichosis can discharge and anchor the animal to a firm substratum when it feeds on bacteria. So this type of trichosis can take part in anchoring of this animal on the firm substratum. And some other believes that these are organelles of defense. Some says that this particular structure is the organ of defense. Then about the next important structure, that is a nucleus. Paramecium is a heterokaryotic organism. Paramecium is a heterokaryotic organism as it possesses or having two types of nuclei. Paramecium have two types of nuclei. So this is a heterokaryotic type of organism. There is having one large nucleus. That large nucleus is called as macronucleus, and also having a small nucleus, and this small nucleus is called as micronucleus. This macronucleus is a kidney-shaped structure. Macronucleus is a large nucleus and having a somewhat kidney-shaped structure and possess many nucleoli. Under this structure in macronucleus, there is having many nucleoli and having a chromatin material in the form of DNA. So all these chromatin material and nucleoli are present in this macronucleus. While micronucleus is lodged in a depression. This micronucleus is lodged or found in a depression on the surface of the macronucleus. It is a spherical structure with diploid number of chromosomes, and this micronucleus control the reproductive activity of the organism. Micronucleus controls the reproductive activity of the organism, and macronucleus take part in the metabolic activity of the cell. Day-to-day metabolic activities can be controlled with the help of macronucleus, and micronucleus take parts in the reproductive activity. Then there are having some contractile apparatus. There are two contractile vacuoles. That is, one contractile vacuole is found towards the anterior side, that is called as anterior contractile vacuole. There are two contractile vacuole having a somewhat fixed position in the endoplasm. One vacuole lies near each end of the body close to the dorsal surface. Each of them is surrounded by particular type of tubes that is called as radial canals. So here is a contractile vacuole, and this contractile vacuole can be supported by elongated tubes that is called as a radial canal. And these radial canals are about 6 to 10 long, narrow, spindle-shaped radial canals are found towards this particular inner structure of the anterior contractile vacuole. Like that of the anterior contractile vacuole, there is having another vacuole which is found towards the posterior side, that is called as posterior contractile vacuole. So they are having two contractile apparatus, that is anterior contractile vacuole and posterior contractile vacuole. And each contractile vacuole opens to outside through a permanent pore which is found or formed in pellicle. Then there is again having a structure which is called as cytophage. This cytophage, which is found near the posterior end of the body, cytophage is found towards the posterior end of the body. Particular area here we can find the cytophage or cytophage. This is found near the posterior end of the body, and which is little behind the cytosome, near to the cytosome. Little behind to the cytosome, this cytophage structure is found. Then this cytophage is a small, which can occupy a small portion of ectoplasm, and pellicle is here where the cytophage is found. Here the pellicle is weaker than all over the body, and here at the time of egestion a minute aperture called as cell annus is formed or visible. So at the time of removal of particular material, at the time of egestion, here on the pellicle there is forming a particular structure that is called as cell annus, and this is about the cytophage. Then when we see the TS of cilia, this cilia, when we see under electron microscope, has shown that each cilium consists of a particular structure, fundamental structure. It consists of a fluid matrix. There is having central area, it is called as matrix. This particular area which is called as central matrix, and surrounded by outer membranous cyst, and this matrix is surrounded by outer membranous cyst, outer membranous sheath, which covers the matrix part of that particular cilium, which is continuous with the outer cell membrane of the body. Within the matrix there are having particularly 9 peripheral fibers. Within the matrix there is having 9 peripheral fibers, peripheral longitudinal fibers, which runs along the whole length of the cilium body. Each fiber is formed by 2 subfibers. Then each fiber is formed by 2 subfibers. These fibers are made by 2 subfibers, and one of which is carries an double row of short arm or projection, and all this, and there is having one incomplete arm-like structure, incomplete fiber, that is having a pair of short arms or projections, and all that runnings in the same direction. All that fibers, that peripheral fibers are running in a same direction, like a clockwise. And between the central and peripheral fibers, there is having accessory fibers. Central fibers in between the in between the central and peripheral fibers, having 9 additional accessory fibers, or also called radial spokes. There is having 9 radial spokes. These 9 radial spokes are attached to the peripheral fibers, or which connects the central fiber with the peripheral fiber. Central fiber connected with the peripheral fibers by this particular type of radial spokes. And this is all about the TS of cilium, or the TS of cilia. The TS of cilia having matrix, and this can cover by outer sheath. There is having peripheral fibers, 9 peripheral fibers. In that 2, they are having each peripheral fibers having two complete subfibers and having a pair of arms. Again, there is having the central fibers having a pair, and the central fibers connected with the peripheral fibers by the particular type of radial spokes. So this was about the structure of paramecium and TS of cilium. So thank you for watching this video, and if you like this video, then share with your friends.